You are very much interested in the science of what's going on here. I want to ask you about what you're doing with the EACR ESMO initiative, but first, a little bit about your own work, because you're a brain cancer specialist, molecular mechanisms are your bag. What to you is the most exciting thing that you're working on at the moment?
So we are making a lot of effort, or investing a lot of efforts to try to understand the molecular mechanisms behind brain tumors. And what we want to find is new therapeutic targets, but also we want to find biomarkers to help us predict responses to particular treatments. And to do so, what we do is we try to analyze and study both the samples from the patients undergoing surgery, but also we generate models to reproduce those tumors.
What do you think are the most exciting biomarkers?
So, well, right now we are thinking that maybe circulating biomarkers could be a way to address the intratumoral heterogeneity that all tumors have, but particularly in brain tumors, in glioblastoma, is very, very high. So we think that by looking at maybe ctDNA or even CTCs, we could see what are the characteristics of the tumor that a patient has and how to treat that patient optimally.
Now we have a huge cancer meeting here at ESMO in Madrid. What for you are some of the highlights coming out of that area, circulating biomarkers?
Yes, well, I think here there is a lot of excellent talks on circulating biomarkers, both on ctDNA, circulating tumor DNA, free DNA in the blood, in the plasma, but also CTC, circulating tumor cells. And both ways might be a way to really better diagnose patients and better find the right treatment.
How much progress has been made? I'm thinking of clinicians wanting to use these techniques. How much progress has been made so far?
Well, the progress is tremendous, although we are still far away for a really well-defined methodology to do this type of things. But we know that there is ctDNA, circulating tumor DNA. We know that that DNA is recapitulating the intratumoral heterogeneity, all the alterations that that tumor has. And we know that this can be monitored in time in response to treatments. So we have to work much more on that, but we have the first indications that this could be a really nice way of monitoring tumors.
Mm-hmm. Is there any indication yet that you could predict metastasis early and therefore do something about it?
Well, it's still early days in this sense, but we have indications that you can predict prognosis based on this circulating. So little by little we will get there.
Mm-hmm. Another very vital area here at the moment I know is immunotherapy. Mm-hmm. What kind of promising things do you see coming up at this conference in that?
I think here in this conference what is very relevant is to try to think about combination of different immunomodulators or even immunomodulators with other targeted therapies. I think this is a way that people are thinking that could be very interesting in this sense.
Mm. I must ask you about something that you're heavily involved with here, and that's the Young Oncologists Meeting that's just happened, and that's between EACR and ESMO, this initiative. What's been happening?
Well, I am a member of the executive committee of the EACR, of the European Association for Cancer Research. And through the contact with ESMO and the Young Oncologists, what we want to get is do a kind of an integration of the young scientists and the young clinicians, doing something together to learn from each other and trying to integrate and do synergisms to do translational research together. So what we did yesterday was to generate a workshop showing how clinicians and basic scientists can be together learning from each other.
Mm. So what progress do you think has been made in that direction?
Well, here the progress is a question of contacting, learning. It's important that the basic scientist understands clinical science and clinical scientists understand basic science. And it's a question of explaining what each other does and working together.
Now, bridging that gap between the bench scientist and the clinician is a difficult one. There's a lot of talk about bench to bedside. What's your clue to the secret of success?
Well, the clue is to getting together and talking, trying to learn each other languages so they can merge and work together. Comprehensive cancer centers where bench, bed are all integrated. Projects should be together. this is the way to go. So young oncologists and young scientists, which are young, they can get together and begin to learn how to generate these bridges.
So what message would you like to leave cancer doctors and cancer scientists with from this recent experience you've had?
So I think for the cancer doctor it's very relevant to understand the molecular mechanisms of tumors, while a basic scientist doing biomedicine of translational research has to understand the way to deal with patients. So my message is get together, talk, collaborate, and do synergisms in this sense.
Thank you very much.
Thank you.